All right, so what I want to talk to you about today is different ways of depicting molecules, what we call different kinds of molecular models. And I also want to talk about color schemes. How is it that we color atoms and molecules? And what are some of the approaches that are used for choosing colors for these things? Um, I know I'm going to sound like a broken record. I've said this 100 times already, but atoms are smaller than the wavelength of visible light, which means that visible light cannot reflect off of the surface of individual atoms. Therefore, you can't see atoms. The whole concept of what an atom looks like is completely irrelevant. It can't look like anything since light can't reflect off of it. So that means that atoms cannot have color. The different colors are the different wavelengths of visible light: red, orange, green, yellow, blue, violet. These are all different wavelengths of visible light. Since visible light can't reflect off an atom, it can't have a color. Atoms can't have shadows. They can't have reflections. All of these things would depend on light either reflecting off of the surface or the atom blocking the light to create a shadow. So when we create an illustration of a molecule, we are not creating a picture of what the molecule looks like. The whole concept of what a molecule, quote unquote, looks like is irrelevant because it can't look like anything because light can't reflect off of it. Instead, when we create an illustration of a molecule, we're attempting to depict something about its three-dimensional structure. We're trying to show the position of the individual atoms in three-dimensional space, the relationship of individual atoms to one another in three-dimensional space, the angles of the bonds that are formed between neighboring atoms, and in some cases, we may represent the size of each individual atom by its van der Waals radius, which is essentially a measure of the size or the diameter of the atom. And if we look at the van der Waals radii of the individual atoms in a molecule, it can give us a sense for the shape of the overall 3D surface of that molecule. Likewise, remember that atoms can't have color. So the colors that we use to represent individual atoms are meant to distinguish the different elements from one another. So we use different colors to distinguish carbon from hydrogen, from oxygen, from nitrogen, from sodium, and so forth. These are not the actual colors of the atoms because the atoms can't have color. There are other color schemes. So the one very common color scheme is to just colorize the individual elements to distinguish them from one another. And we'll talk about there's kind of a standard that's been adopted in chemistry for coloring atoms different colors and coloring the elements different colors. But there are other color schemes as well that are meant to depict some different feature of the physical properties of an atom or molecule. So there are lots of different kinds of models, lots of different ways of representing molecules, and there are a handful of different color schemes, a handful of different approaches to choosing the colors for atoms and molecules. And it's important to remember that one of these approaches, one kind of model or one kind of color scheme, is not always the best choice. One of them is not inherently better than another. We use whichever type of model and whichever color scheme is going to show the specific properties of the molecule that we're most interested in. So are we most interested in knowing what all of the different atoms are in a molecule? Do we want to be able to distinguish the carbons from the hydrogens, from the nitrogens, from the oxygens, and so forth? In which case we might choose one color scheme, but we might use a different color scheme for something else. And we may use any different kind of model depending on the physical properties of the molecule that we're trying to represent. So what I want to do today is walk you through these different kinds of models and talk about the advantages and disadvantages of each kind of model. When would it be appropriate to use one kind of model versus using a different kind of model? And then do the same thing talking about color schemes. So believe it or not, these things are all ways of representing molecules. These are all considered types of molecular models. Even something as simple as a chemical formula, in a sense, is a way of representing some physical property of a molecule. What the chemical formula tells us is the relative proportions of the different kinds of atoms that make up a particular molecule. So we know that H2O means it's a molecule that consists of 2 atoms of hydrogen and 1 atom of oxygen. And C5H12, that would be pentane. That's 5 atoms of carbon and 12 atoms of hydrogen. So we know instantly in looking at one of these chemical formulae, we know exactly what the relative proportions of the different atoms are that are present. These chemical formulae have been around since the early 1800s. The concept of the atom was first put forward in about 1805. And then shortly thereafter, the early chemists started to actually, run experiments where they could actually determine the relative numbers of atoms in a different compound. So they were able to determine very early on that water consisted of 2 parts hydrogen, 1 part oxygen, that methane was 4 parts hydrogen, 1 part carbon, and so forth. So they started drawing these chemical formulae very early on in the 1800s. But one thing that they didn't know yet in the early 1800s is they didn't know which atom was actually attached to which other atom. They knew that water was 2 hydrogen atoms and 1 oxygen atom, but they didn't know, is the oxygen in the middle and the hydrogens are on either side, or is the oxygen at one end followed by 2 hydrogens in a row? They had no idea in the early 1800s. But by the middle 1800s, they had started to determine that. They actually knew which atom was bonded to which other atom. So we see a new kind of model start to appear in the scientific literature, beginning in, maybe the 1840s or thereabouts, maybe 1850, somewhere in that neighborhood. This is known as a structural formula. And you can see from the picture, this is just like the ones I had on the quiz today. We can actually tell that these carbon atoms are all bonded to one another by the presence of the little black lines representing bonds. And we can tell that the hydrogens are attached to specific carbons in the chain. And there's a single OH here that's attached to one of these carbons in the chain. So we can tell exactly how many atoms are present in this molecule, and we can tell which atom is bonded to which other atom. The structural formula is sometimes known as a Kekulé diagram. This is actually named after a guy that I mentioned on Monday, the guy whose name I just enjoy, Friedrich August Kekulé von Stradonitz, who usually just goes by Kekulé. And he was the first chemist in the mid 1800s to publish an illustration like this, using these little sticks to represent bonds. Turns out there was another chemist in England who actually beat him to it. But the problem was, I think he was a grad student and his thesis advisor delayed the publication of his thesis. And so Kekulé beat him to the punch. But this other guy named Cooper, Archibald Cooper, had actually used it earlier, but it doesn't matter. People call it a Kekulé diagram now. There's something similar to a Kekulé diagram known as a skeletal diagram. And this also shows which atom is bonded to which other atom, but it uses a little bit of a shorthand notation. I talked about this briefly last week, that sometimes you'll see a picture of a molecule that doesn't actually show you all the atoms. The atoms are implied, but they're not shown explicitly. So we would say that they are implicit atoms, but they're not explicitly shown. So a lot of the atoms are shown here, like there's a phosphorus right there. There's a bunch of oxygens and some hydrogens. There's another oxygen up here, a bunch more oxygens. There's a couple of nitrogens shown in here. But what's missing are all of the carbons. And if you remember from a week or so ago, I mentioned that every single corner point, so every corner where 2 lines intersect, if there's nothing else there, if there's no other letter there, that means there's a carbon. So that means that this ring-shaped structure actually consists of 6 carbons arranged in a ring, 1 carbon at each of those corners. There's another carbon up here at this sharp point. That corner point is going to represent a carbon. And then we've got 6 more carbons in a ring here. And then all of these long zigzagging tails are long chains of carbon atoms. Every one of the corner points where 2 of the bonds join, that's another carbon atom. the endpoints of black lines also represent carbons. So every corner point and every endpoint is also a carbon. So what that means is that this little stick up here on the right side of that ring is also a carbon atom. So there's a carbon out at the end of that stick. And then at the very tips of all of these little tails, there are more carbon atoms at the very end of each of those black lines. So all of the carbons are hidden. Yeah.
And then doesn't that part the hydrogens also bonded to the carbons?
Exactly. That's the next thing I was going to say, is that the other thing that's missing are most of the hydrogens. Some of them are shown. Like whenever you have an OH group, hydroxyl group, those hydrogens are shown. These nitrogens, we depict, one extra hydrogen, attached to the nitrogen there. Some more OH groups down here. But every one of those carbons that is hidden probably also has some hydrogens attached to it, because remember that the valence of carbon is 4. Carbon always forms 4 bonds. So if you only see 3 bonds connected to a carbon, it means the 4th bond has to be filled with a hydrogen. So, for example, if we look at that corner point right there, and I don't know if well, I was going to say let me use my Wacom tablet, but there's no pen. Oh, there it is. Let's see, is this, yeah. If I get my little highlight, I think I kind of overused the highlighter a little bit last time, but this would be a good place to use it. All right, so there's a carbon right here at that corner point. And we can see that it's bonded to that oxygen, that's 1 bond. It's bonded to a neighboring carbon up here, that's 2. It's bonded to another neighboring carbon down there, that's 3, but that's only 3 bonds. That carbon has to have a 4th bond, and that's going to be attached to a hydrogen, which is hidden. This carbon right here is bonded to one neighboring carbon and another neighboring carbon. That's only 2 bonds. So that carbon must have 2 hydrogens attached to it. Same thing up here. This carbon, it has a bond to that oxygen. It has a bond to that ring, but it must also have 2 other hydrogens to fill in the valence. And there's a whole bunch others missing that I'm not going to try to fill in, but moving down to these long tails, there's a carbon here at that corner point. It's bonded to 2 of its neighbors. That's only 2. So that means there must be 2 more H's out here. And all of these endpoints are also carbons, including this endpoint up here. Well, that only has 1 bond. It has a single bond attaching it to the rest of the ring. That means there must be 3 more hydrogens out there. Same thing down here. Every one of these is going to have 3 hydrogens attached to it. Okay, so these skeletal diagrams are kind of like Kekule diagrams, but they hide a lot of the atoms, because you can just imagine how cluttered that picture would be if we tried to draw in every single carbon and every single hydrogen in that. So the skeletal diagrams are nice. Kekule diagrams and skeletal diagrams are both useful because they show us how many atoms are present. They tell us exactly which atoms are bonded to which other atoms. But one thing that structural diagrams or structural formulas and skeletal diagrams don't show is they don't make any attempt to try to show the 3-dimensional relationships between these atoms. We really don't have any good sense for how these things are organized in 3-dimensional space. And it wasn't until the latter part of the 19th century, the late 1800s, that chemists began to get some sense for the 3-dimensional arrangement of atoms in space. And really it wasn't until the 19-teens, with the development of X-ray crystallography, that we really started to be able to understand how atoms are arranged in 3-dimensional space. So then we started to see some other kinds of models appear, such as the wireframe model or the line model. And this is showing all of the atoms. I mean, they're not labeled, but you have to use the same principle to understand what's what. Now, this is actually showing the bonds between atoms. There are carbons at the corners of this ring. In fact, the dotted line tells me that that's a benzene ring with alternating double bonds going around there. These others are not benzene because they don't have the double bonds in them. These would be cyclohexane rings, six-sided carbon rings with just single bonds. All of these little sticks here are actually bonds to hydrogen atoms. You just don't see the hydrogens either. The red indicates oxygen. So there's an oxygen atom out here that's attached to this benzene ring. There's another oxygen atom out there. And there's a little subtle suggestion of how these things are arranged in 3D space. It's a little hard to tell from this figure, but it actually uses a little bit of what's called depth cueing, where things in the background are actually a little bit more faded and things that are closest to the viewer are a little bit more solid. It's a little hard to tell in this particular picture that there's any depth cueing going on. I almost never use a wireframe picture. I can't really imagine any time that you would want to use one of these things. There's a variation on the wireframe, which is actually a lot more useful and a lot easier to understand. That's the stick model. So the only difference here is that the wires are fat, right? And instead of just being thin little lines, we've added these thicker cylinders to represent the bonds between the atoms. And now you start to get a much better sense for the three-dimensional arrangement of these atoms that make up this molecule of estrogen. And you can see that depth cueing where things that are going off into the distance are a little bit lighter and things that are coming towards us are a little bit more crisp and a little bit more, have a little more contrast in them. You can also see that each of those little sticks actually represents a bond between two atoms, and the color tells me what atom it is. So there's a carbon here that's part of the benzene ring, and it's bonded to a hydrogen out there. That little white stick means that there's a hydrogen out there. And so all these little white sticks represent hydrogens. Here's, this is actually an OH group, a hydroxyl group. So there's an oxygen at the center of these two bonds, or at the corner of these two bonds, and then bonded to a hydrogen out here at the end of that little white stick. And again, you see how the oxygen atom is forming a bent configuration. Same thing over here on the left. There's an oxygen at that corner point, and the two bonds attached to that oxygen are forming that bent configuration. Here's a carbon coming towards us. This would be a methyl group. This would be CH3, which is then attached to the rest of the molecule. So there's a carbon in there and then three hydrogens. And notice how all four of those bonds are arranged in that tetrahedral geometry with equal angles of 109.5 degrees. Another slight variation on the wireframe and stick model is the ball and stick model. The only difference here, it's got the same sticks, the same three-dimensional tubes, but now we've added small spheres to represent the location of the atoms that are bonded to one another. Notice that all of the atoms are the same diameter. So even the white hydrogens are the same diameter as the dark gray carbons and the red oxygens. That's because in this type of ball and stick model, we're not really trying to represent the true size or the true diameter of the atoms. We're not using that van der Waals radius to represent the diameter of the atoms. Every atom is identical in size. But all three of these models, the wireframe, the stick, and the ball and stick model, these all do a very good job of showing us which atom is bonded to which other atom and how the atoms are arranged in three-dimensional space. There's another kind of model, which is very common. And again, it's going to be the exact same molecule. All of these are estrogen, just they're all viewed from the same angle at the same size, just different kinds of models. Here's a different kind of model, which I'm sure you've seen these. The little plastic models that I handed around last week were called examples of this kind of model. This has several names. It could be called a space filling model. That's a very common name for this type of model. It could also be called simply a sphere. model. Some of the molecular visualization applications that we're going to use refer to it as a sphere. It has a third name. It's also known as a CPK model, which we'll come back to and talk about that more in just a minute. With these sphere models or space filling models or CPK models, we're actually representing every atom by its van der Waals radius, which in a sense is the three-dimensional space that the atom takes up. It's the space occupied by the cloud of electrons around the atom. When atoms are covalently bonded to one another, those van der Waals radii actually overlap. So this benzene ring right here, which are 6 carbon atoms bonded together, notice how the carbons are all intersecting and overlapping one another, because these carbons are all sharing electrons because of the fact that they're covalently bonded. And then this oxygen over here is bonded to one of the carbons, and their spheres overlap because, again, they're sharing electrons to create that bond. The oxygen is overlapping with the hydrogen because the oxygen and the hydrogen are sharing electrons. So we use the van der Waals radius to represent the diameter of the atoms, but they always overlap if they're bonded to one another. Now, there are some pros and cons to a space filling model. I mentioned before that every model has its uses and its disadvantages. One advantage to a space filling model is it starts to give you a sense for the overall three-dimensional shape, the overall shape of the surface of the molecule, which can be very important sometimes, especially in a few minutes, we're going to talk about proteins. The overall three-dimensional shape of a protein is very important to understanding its function. So space filling starts to give us a sense for that overall three-dimensional shape. One of the downsides to the space filling model is sometimes it's hard to tell which atom is bonded to which other atom. It's not so bad over on the right. I can tell that that's a ring of 6 carbons that are all bonded to one another. I can tell which carbon these hydrogens are attached to. I can tell that that OH group is attached to a certain carbon. But you get over here on the left and things start to get kind of a jumble, and it can be a little hard to tell which atom is attached to which other atom because they're all overlapping and intersecting. So that can be a downside. If it's important to see the full structure of the molecule and understand which atom is bonded to which other, space filling may not be your best choice. So these are the van der Waals radii of many of the atoms. It's not the full periodic table, but it's a lot of it. And it's organized as a simplified periodic table with column 1, column 2, column 3, indicating how many electrons are in the outermost shell. And then we have the horizontal rows indicating how many shells are present. So the top row is only the inner shell with 2 electrons. And then we add a second shell, third shell, fourth shell, and so forth. And we've talked about this before, about how the van der Waals radius varies. As you go down the periodic table, the van der Waals radii get progressively bigger because you keep adding more shells of electrons to the outside. But also, as you go across the periodic table from left to right, the atoms are bigger on the left and they get smaller as you go to the right. Even though you have more electrons in the outermost shell, you've also got more protons in the nucleus, so there's a stronger electrical attraction between the positively charged protons in the nucleus and the negatively charged electrons orbiting. So the electrons are actually held tighter, closer to the nucleus. So that's why they get smaller as you go from left to right. So you can easily find a picture like this in a Google search if you ever need to try to guess about how big an atom is going to be if it's part of a molecule. All right, so I mentioned that the space filling or sphere models have a third name. They're also known as CPK models. And that's actually named after 3 people who were instrumental in developing this type of model, this type of representation. And one of the key players is my buddy Linus Pauling, who I have mentioned at least once before. He's the guy who wrote that book, The Nature of the Chemical Bond, if I'm remembering that correctly. Basically applied quantum mechanics to chemistry to determine how bonds happen. And that was in the 1930s, and he won his first Nobel Prize for that. after he had won his Nobel Prize for solving how chemical bonds work, he shifted his attention to something else, and he's the one who discovered the concept of the van der Waals radius and also measured the van der Waals radii of many of the common elements. And when he realized that atoms had kind of a predictable size, a predictable diameter, he decided that wouldn't it be cool if you could actually make little wooden models where there were a whole bunch of little balls, a whole bunch of little spheres, and the diameter of those spheres was the van der Waals radius of that particular element. So he was the first one to invent the kind of model that he's holding in his hand, which was the space-filling model. And so not only conceptually to develop, but he literally helped develop the first physical 3D models that were then manufactured and sold to chemistry students so that they could play around with them and learn some chemistry. So he's holding a set of these models. I can't tell you what that molecule is exactly. I can tell it's got a benzene ring in it, but I can't tell what else that is. So Linus Pauling was at Caltech, and he was working closely with a colleague named Robert Corey. And it was actually the two of them together who devised this style of molecular model. And so here's another picture of Linus Pauling on the left, real spiffy dresser, with his colleague Robert Corey at Caltech. And they're playing with some of these space-filling models. They've actually got a model here, which I have a picture of, I think, somewhere in this lecture, of this is actually known as an alpha helix, which is an important component of proteins. And they had constructed it using these models. So originally developed by Robert Corey and Linus Pauling, so that's the C and the P in the name. But then in the early 1960s, there was a chemist working at NIH in Washington named Walter Colton, and his contribution was to really just create a different mechanism for connecting the spheres together. So these were known as Colton connectors. And I actually found this old ad advertising a new type of molecular model available from some company. And it says, new Corey-Pauling space-filling atomic models with Colton connectors. And from that point on, these became known as CPK models. So that refers to the physical models made out of either wood or plastic that you can put together in chemistry class. It refers to any representation, any space-filling representation of a molecule. And later on today, we'll talk about the fact that it's also a term that's used to describe the color scheme, where you use different colors to distinguish the elements. These space-filling or CPK or sphere models are very popular with artists because the large spheres lend themselves to fancy rendering in your favorite 3D program. Now, this one on the left is not particularly fancy, but it is a CPK model of a cholesterol molecule. It's actually rendered directly in one of the molecular visualization programs that we're going to use in this class. Here's one I built of DNA, rendered in an older version of Cinema 4D. And, every atom represented by a sphere of its van der Waals radius. The yellow ones are phosphorus, and they're noticeably bigger than most of the other atoms in there. And here's one. I rendered this in Cinema 4D quite a few years ago, just having fun, playing with reflections and stuff. But of course, this is all bullshit, right? They can't have foreshadows and shiny reflected, shiny highlights. They're certainly not going to have these kinds of internal reflections that make these things look like, a bunch of Christmas ornaments. So even though sometimes we artists like to have fun and kind of push rendering to the extreme, just, remember that it's not real, right? Atoms don't really look like that because they don't look like anything. Here's another example of a space-filling model or sphere model or a CPK model. This time we're looking at an entire protein called lysozyme, which is used to break things down inside the lysosome sort of digestive enzyme inside cells. And the CPK model or space filling model kind of gives us a sense for the overall shape and structure of the protein. But if you remember, proteins are amino acid chains, right? It's simply a linear chain of amino acids that's produced at the ribosome during the process of genetic translation. And if you're studying protein structure, sometimes it's very, very important to actually understand how the amino acid chain folds to create the compact blob that you see in front of you right now. So this sort of space filling or sphere model of a protein, like I said, it's pretty good if you want to understand the overall three-dimensional structure of the protein, the structure of its three-dimensional surface. But this would be a terrible choice if you wanted to understand how the amino acid chain actually folds to create this model. So turns out there are a couple of special kinds of models that are used specifically for representing proteins. And two of these kinds of models focus on being able to visualize the folding of the amino acid chain as it kind of winds around inside the protein. So in order to understand these two other kinds of models for visualizing the folding of the amino acid chain, we do have to learn a little bit about proteins. We'll come back to this later in the course. I got a whole lecture for you about proteins, but for now, let's just review a little bit about protein structure, and hopefully this is all review from A&P or you know, your human bio or intro bio course. So all proteins are made up of small subunits called amino acids. So here we see two amino acids. And in one of the lectures, I think on Monday, I talked about what exactly is an amino acid. It has a carbon at the center known as the alpha carbon. That carbon is attached to an amino group, which is NH2. And that alpha carbon is also attached to a carboxylic acid group, which is COOH, carbon with a double bonded oxygen and an OH group. And it's the fact that amino acids have both of these things that's what gives it its name. It's a molecule that has both an amino group and an acid group, hence the name amino acid. Now that alpha carbon also typically has a hydrogen attached to it, and then it has something represented by the letter R here, which is the side chain. That R is variable. There are 21 amino acids that are commonly found in living things, and they all differ from one another by their side chains. So there are some very simple amino acids, like the simplest one is glycine, where the side chain, that R, is literally just a hydrogen, nothing else. There's alanine, where the R is a methyl group, CH3. But then there are some very complex amino acids. Phenylalanine has a carbon there, but then it has an entire benzene ring attached in that R location. Tryptophan is another one that also has a big ring structure in its side chain. There are some amino acids that even have sulfur in their side chain, methionine and cysteine, two amino acids that actually help to create the three-dimensional structure of proteins. So that side chain can be very small, it can be very big, but it's variable, and that's what distinguishes one amino acid from another. Right, so you start out with these individual amino acids, and these things are carried by the transfer RNA molecules, the tRNA, and delivered to the ribosome. And the tRNA molecules hold these amino acids next to one another very closely in a very specific configuration, so that the OH group of that carboxylic acid, of one amino acid, is held right next to the amino group of the next amino acid in line, and a chemical reaction occurs where the OH from the carboxylic acid breaks off, one of the hydrogens from the amino group breaks off, they join together to form HOH, better known as H2O, or water, and that water molecule is expelled, just goes out into the cytoplasm. And the carbon of the carboxylic acid is now bonded directly to the nitrogen of the amino group on the next amino acid. So we now end up with a bond known as a peptide bond, and the molecule that we produced is now known as a dipeptide because it consists of two amino acids stuck together. So we have the one amino acid over here, and then the carbon from its acid group bonds directly to the nitrogen of the amino group of the next amino acid in line. And then the same thing happens again. These two move over, another tRNA comes in delivering another amino acid. It holds that amino acid right next to the OH group of the dipeptide, and another peptide bond forms where the amino group of the next amino acid then joins that carbon, and it continues to elongate that way. You have new tRNAs coming into the ribosome, delivering new amino acids and constantly making this polypeptide chain longer and longer and longer. So once you get a whole bunch of amino acids strung together, we call that a polypeptide. Now, technically, the definition of a protein is it's a polypeptide chain that consists of 50 or more amino acids. But that's not entirely true because there are actually proteins that have fewer than 50. So really a protein is really just a string of amino acids that has some function, regardless of how long or how short it is. Now, a couple things to notice about what happens when you string all these amino acids together. For one thing, you get this repeating pattern of nitrogen, carbon, carbon. That's all part of one amino acid. And then that's attached to the next one, nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, and so on, running the entire length of the protein. Those atoms bonded together make up what we call the backbone of the protein. You can almost think of it as being like the skeleton of the protein. And if you want to be able to understand how that amino acid chain folds to fold up the protein, to create a compact three-dimensional structure, it's very helpful if you can look only at that backbone and not see anything else. So in a second, I'll mention to you that there's a type of model known specifically as a backbone model of a protein, which looks only at the nitrogen, carbon, carbon, nitrogen, carbon, carbon, only the stuff that's highlighted here in green, and it hides everything else. It hides the hydrogens, it hides the oxygens, and most importantly, it hides all of those large complex side chains. Right? So the side chains are completely hidden. But that's a great way to be able to visualize the folding of the amino acid chain inside the protein molecule. The other thing to notice is that the very first amino acid that is deposited at the ribosome starts with its amino group at one end, and then new amino acids are always added to the carboxylic acid side of the previous amino acid. So when I want to add another amino acid to this dipeptide, it would never get added over here on the left. The new amino acids are always added over here at the carboxylic acid end. So that means that every protein has a beginning and an end. The beginning would be this very first amino acid that was deposited at the ribosome, and it begins with its amino group. So the start of the amino acid chain, the start of our polypeptide or protein chain, is known as the N-terminus, because it's that amino group of the very first amino acid that's deposited at the ribosome. And then all subsequent amino acids are added at the C-terminus. So the very last amino acid that gets added when translation is done, very last amino acid that gets added to that protein is going to end with its carboxylic acid group. It's going to be added over here at what we call the C-terminus. And some of the ways of depicting the folding of the amino acid chain will actually show you that direction, show you that directionality from the very first amino acid, which is the N-terminus, to the very last amino acid that was, deposited at the ribosome. That would be the C-terminus. All right, so here is a picture of that exact same enzyme, lysozyme, but viewing it only as a backbone, where we're looking only at nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon. Every one of these little short straight segments represents the nitrogen, carbon, carbon of an amino acid. And you can actually follow the folding of that backbone fairly easily. It gets a little hidden back there, but I think it goes up this way, zigzags around, comes back down, comes over here and around. I don't know, losing it back there a little bit. Things get a little topsy-turvy back there, but theoretically you should be able to follow that amino acid chain from beginning to end all the way through. Now this particular picture doesn't actually identify the N-terminus and the C-terminus. So I don't actually know if it started here at this end or if it started at that end. So I actually don't know the directionality of this, but we'll see in a few minutes that there's some other models that actually do show that. So this is literally the same as that. Exact same protein, same scale, same magnification, same view, same angle, just two very radically different ways of representing the same molecule. And in this space filling model, we're seeing all the side groups, all the R groups. Whereas in the backbone model, they're hidden, and we're only seeing that backbone. All right, a little bit more about protein structure. To understand the next kind of model, you have to know a little bit more about the structure of proteins. So every protein can be described as having either 3 or 4 levels of complexity or levels of organization. So every protein is going to have what we call a primary structure. That is nothing more than just the list of amino acids in a straight line in order. And they're going to be listed in the order that they were deposited at the ribosome. So you're going to start with the very first amino acid, which is usually methionine, by the way. That's usually the very first amino acid that's created by the start codon during the process of translation. And then you're simply going to list in order every amino acid. That's the primary structure, just a straight chain of amino acids. Every protein also has what we call a secondary structure. As soon as that amino acid chain is created at the ribosome, it begins to fold spontaneously. And sometimes it folds into a little corkscrew, a little spiral, known as an alpha helix. Other times the amino acid chain forms like a little hairpin turn and kind of folds back on itself, and then forms another hairpin turn and folds back on itself, creating a flat sheet of amino acids known as the beta sheet. And there are other kinds of little folds and things that have special names to them. These spirals, the corkscrew spirals, and these sheets of amino acids, these are all elements of what we call the secondary structure, where that linear chain of amino acids begins to initially fold itself spontaneously into spirals and sheets and other kinds of folds. Every protein also has the third level of structure, known as tertiary structure or third degree structure. This is where all of those secondary structures, all of those spirals, folds, sheets, and other elements get folded even more to create a compact three-dimensional blob. That is the tertiary or third degree structure of a protein. Now, there are a lot of proteins that are functional at this point. Something like myoglobin, which stores oxygen in muscle tissue. That has a primary structure, secondary structure, and third and tertiary structure and that's it. It's now a fully functional protein. The sodium channel that allows sodium ions to pass through the cell membrane, that has just a tertiary structure, and that's it. But there are other proteins that actually have to have an even higher level of structure before they're functional. This is what's known as a quaternary structure or fourth degree structure. This is when you have a protein that's actually made up of multiple different amino acid chains. And each of those individual amino acid chains has primary, secondary, tertiary structure. But then those multiple amino acid chains then have to join together to form an even larger, more complex structure. And that would be a quaternary structure. So the classic example of a protein with quaternary structure is hemoglobin. Hemoglobin is made up of 4 separate amino acid chains. 2 alphas, and a delta, I think I have a picture of it that'll remind me. But so 4 separate amino acid chains that are synthesized independently on the ribosome. Each one folds into a compact blob, but then those 4 blobs come together to form one functioning hemoglobin molecule. So that would be an example of quaternary structure. So we've already seen this picture. This is just showing the primary structure. It's that linear chain of amino acids simply joined together end to end. As soon as that amino acid chain is formed, it begins to fold spontaneously to form these things called secondary structures. And this is one of the most common secondary structures, known as an alpha helix. And so what's happening here is you've got the amino acid chain folding into a corkscrew, into a spiral. So the blue circle would be a nitrogen. The carbons would be represented by gray. So we've got nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, and so forth. That's the backbone of the protein, and that backbone coils into this helix, known as an alpha helix. Now notice the little dotted lines, the little black dotted lines. Those are what are known as hydrogen bonds. It's not really an ionic bond. It's not really a covalent bond either. It's a weak attraction between atoms of opposite charge. So, for example, here we have a hydrogen, which has, even though it's not an ion, but it's still a little bit more positive than the rest of the molecule. And then over here is a double bonded oxygen, which is, again, it doesn't have a full charge, but it's a little bit more negative compared to the rest of the molecule. Those hydrogens and oxygens are actually attracted to one another. The slight negative charge of the oxygen is attracted to the slight positive charge of the hydrogen. And that creates this weak attraction. And it's these hydrogen bonds that actually hold the alpha helix in shape. And we'll come back and talk about this more when we talk about proteins. Oh, a little hint. The structure of the alpha helix was determined by my buddy Linus Pauling. Here's another very common element of secondary structure known as the beta sheet. So what happens in a beta sheet is you've got your amino acid chain, and it would actually be going from right to left in this case because there's nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, and so forth. This chain would be running basically in a straight line, but then it would make a sharp hairpin turn, which I didn't show in this picture, but there'd be a sharp 180-degree turn. The amino acid chain would then start running the opposite direction, and at a certain point would make another 180-degree turn and turn back the other way. And so this could actually keep zigzagging back and forth numerous times. And these adjacent strands of the amino acid chain are then held together by hydrogen bonds again. Once again, you see the little dotted black lines, and it stabilizes the strands of the amino acid chain into a flat sheet, known as a beta sheet, also discovered by Linus Pauling in the 1950s. So there is a type of molecular model that is used exclusively for proteins that illustrates these different elements of secondary structure. This is what's known as a ribbon model. And in a ribbon model of a protein, the alpha helices are represented as little corkscrew spirals. In this particular protein on the left, we see 3 of them. There's a large alpha helix over there on the left. One down the bottom, there's a little teeny piece of alpha helix kind of hiding in the background. The beta sheets are represented as flat planes, sometimes with arrowheads on them. And the arrowheads are actually pointing in the direction from the N terminus towards the C terminus. So pointing from the beginning towards the end of the protein. And so if you look closely here, you'll see that there's one arrow that comes around this way, and then there's a little loop. That's our 180-degree hairpin turn, and then it turns back the other way. And then we get another hairpin turn and comes back the other way. So we have 3 strands of the amino acid chain that are held to one another by these weak hydrogen bonds to form a flat sheet. And there's a little bit more of it in the back. You see there's a couple more arrowheads in the background. Here's another protein. This one's kind of weird looking because you got all these arrows pointing in different directions. Believe it or not, that is a beta sheet, but the whole sheet is twisted. And then you got a couple of alpha helices as well. There's a big one in the back and a couple of tiny little loops of alpha helix over here. As far as I know, the first ribbon diagram of a protein was actually created in the 1960s. There was a book called what's it called? Structure and Function of Proteins, I think it was called. And I'm drawing a blank on the artist's name. Irving Geis was the illustrator. And he utilized this method of illustrating proteins, but it didn't really catch on. He only used it for a couple of figures. He didn't really illustrate the whole book using these ribbon diagrams. The ribbon diagram was really popularized in the late 70s and early 1980s by Jane Richardson. Jane was a chemist at Duke University down in North Carolina, and she studied protein structure. And in 1980, she was asked to actually write a book. It was actually an issue of a journal, but it ended up being the size of a book. And it was called the Taxonomy of Protein Structure. And basically, she took all of the protein structures that were known up at that time, which in 1980, I think there were only 75 proteins that had been solved in 3 dimensions. But she wanted to compare all of their structures and see if there were any common elements or any similarities between the structures of these different proteins. And so in order to do that, she basically invented this style of drawing. And she actually drew these all herself in pen and ink. These are actually 2 of her drawings from that book from 1980. Now here is a computer-generated ribbon model. And once again, this is the exact same protein shown here and shown here. It's lysozyme from the same view, same scale, same everything, just a different way of representing it. So this is kind of like a backbone model because all we're looking at is the backbone of the protein. We've hidden the side chains, but this particular type of ribbon model emphasizes the presence of these secondary structures. This one is mostly alpha helices. There's a little alpha helix here, another one here, another one in the background, a couple more back there. The only beta sheet in here is way in the back. There's a couple of little strands forming a very small beta sheet. So this is depicting secondary structure, right? So we're seeing the alpha helices and the beta sheets in here, but we're also looking at the tertiary structure of this molecule because we're seeing now how the alpha helices in the beta sheets are then further folded to create a compact blob. Here's another example. This is one that I created in just directly out of a molecular visualization application. This is a ribbon model of a protein called tubulin, which is the building block of microtubules in the cell, that are an important part of the cytoskeleton, the skeleton of the cell. We'll talk about this more when we talk about color schemes, but one nice thing about this ribbon representation is that it actually uses color to distinguish the different elements of secondary structure. So all of the alpha helices are shown in red, all of the beta sheets are shown in turquoise, and then there's a lot of little random pieces of the amino acid chain that don't really have a secondary structure. So all the gray things are just sort of random loops and twists and coils and so forth. The two space filling models in here are not part of the protein. Those are other molecules that are bonded to the protein. I know this one over here on the right with some nitrogens, the blue nitrogens and some yellow phosphorus, that's actually GTP, guanosine triphosphate, which is an energy molecule very similar to ATP, but it's based on guanine instead of adenine. And over on the left are some drug molecules, or actually I think just one drug molecule, and I think it's the drug doxorubicin. It's a chemotherapy drug, but I'm not 100% sure about that. I might be wrong about that. But anyway, so these are a couple of other molecules that I rendered differently because they're not really part of the protein. And then here we see the classic example of a protein that has quaternary structure. So in this example, all of the ribbons are one amino acid chain. It's a single amino acid chain that's folded on itself. But in hemoglobin, we have 4 distinct amino acid chains that are all produced independently at the ribosome from different strands of messenger RNA. And then those 4 chains then have to come together to form a larger, more complex blob. So in orange, we have 2 alpha chains. In purple, we have a beta chain, and in turquoise, we have a delta chain. So 4 separate amino acid chains that then come together to form a single functioning molecule of hemoglobin. And in this case, the space-filling models are actually the heme groups. It's a ring of carbon atoms with an iron atom at the center of the ring. And that's actually what holds the oxygen molecules. Because hemoglobin, of course, is the protein that transports oxygen inside your red blood cells. And it's the iron atom at the center of those heme groups, which is actually what binds the iron and transports the iron in your blood. All right, so we've talked about a couple of ways of visualizing the folding of the amino acid chain inside of a protein. So there's the backbone model, and then there's this very specialized ribbon model that shows the folding of the amino acid chain and emphasizes the presence of these secondary structures in that folded amino acid chain. There's also another type of model that's used for visualizing the 3-dimensional surface of a protein. Now, I've shown you this picture before. This is our space-filling model or sphere model or CPK model of a protein. And it does kind of give you a rough idea of the overall 3-dimensional surface of the molecule. But there is actually another kind of model that's even a little bit better, that does a better job of showing you the overall 3-dimensional shape of a molecule. It can actually be used for any kind of molecule, but it's used especially for proteins. This is what's known as a solvent accessible surface model. And what I've done here is I've drawn a close-up of just a very tiny portion of the surface of a protein. And what you see is we've got a whole bunch of atoms that are all bonded to one another. And, you know, they dip down and they make little indentations and then they come back up towards the surface and they create this complex surface on the outside of the protein. So if you represented this protein just as a CPK or space filling model, you would simply see all of those little spheres representing the individual atoms. But in a solvent accessible surface model, what we're really trying to understand is where are the deeper indentations on the surface of this protein that could serve as binding sites for the substrate that is acted upon by this protein. So you're probably all aware that many proteins function as enzymes, meaning that they promote certain chemical reactions in the body. And they do that by binding to some other molecule. We would call that the substrate. So it may bind to a larger molecule and hold that molecule in a certain configuration and then split a larger molecule into smaller components. Or a protein might do the opposite. It might hold two small molecules close together and encourage those two then to bond together. So a protein, an enzyme can either split something into smaller pieces, or it can combine small pieces together into larger things. And the ability of a protein to do that, to function as an enzyme and to promote these chemical reactions, all depends on the presence of some kind of binding pocket, some kind of pit or dent on the surface of the molecule that's actually going to serve to hold those chemicals together, those substrates together. So the solvent accessible surface is trying to understand where are the little shallow indentations, the small indentations, and where are the bigger ones. And the way it does that is this is all done digitally. This is all done in a computer. Nobody does this on an actual physical 3D model. But the computer software imagines what's called a probe, shown here as this green circle. And it imagines that that probe has a certain diameter to it. And it takes that probe sphere, again, doing this all digitally and in virtual space, it rolls that probe around on the surface of the protein. And there are going to be some places where the probe is in direct contact with the atoms that are close to the surface of the molecule. But there are going to be other places where there are little indentations between neighboring atoms, and the probe can't fit in there. So the probe draws a little surface. And here's a better example. Here we have a somewhat deeper pocket formed between some neighboring atoms, but the probe can't fit in there. So the software actually draws this little green line. It draws a surface showing how far that probe can reach into the interior of the molecule. And what this does is it gives us a new kind of model that shows, kind of smooths out the surface of the molecule, and it only shows where some of the deeper pockets are that might serve as important binding sites for the substrate. Incidentally, the probe sphere in the molecular visualization software that does this, usually you can edit the diameter of the probe sphere, so you can change it. But the default diameter for the probe sphere is typically 1.4 angstroms, which turns out that's the diameter of a water molecule. So what this solvent accessible surface is showing us is where can water molecules actually reach in the surface of this protein. So here is our lysozyme protein again, represented as a solvent accessible surface. So the little patches of blue and red and gray that you see on here and white, those represent the surfaces of the atoms themselves, but then it's drawn this smooth surface. It's almost like it's wrapped the space filling model in a piece of plastic wrap or something, and it kind of smooths out all the little gaps between the atoms. And it shows you where some of the deeper pockets are. Like there may be a fairly deep pocket down here at the bottom, maybe some here in the front. There's definitely a deep pocket up here. So any of those things could serve as binding sites for this protein to interact with some other chemical, some substrate. Here's another example. This is a solvent accessible surface model of a potassium channel protein. And I think I may have brought this with me as an actual model or as a digital model that I can show you later. If I could actually rotate this here and look down at the top, you'd actually see that there's a little pore that runs right through the middle of this protein, which is the actual channel that the potassium passes through. And this would be embedded in the cell membrane of a cell. So there's a little pore that allows potassium to flow either in or out of the cell. Here's an illustration that I created quite a few years ago. This was actually for one of the Netter books. This was Netter's Illustrated Pharmacology. And I was one of the main contributors to that. And this is a pharmacology book, so it's all about the action of drugs. And what they wanted to illustrate was that proteins have these binding pockets on their surface, but that binding pocket may only accommodate certain forms of a particular drug. And many molecules will have what are called enantiomers, which just means different shape configurations of the molecule. A lot of organic molecules have what we call a left-handed and right-handed form. The left-handed form often will have an L in front of the name of the molecule, indicating that it is the left-handed form, also known as the sinistral form, because sinister is the Latin word for left-handed or left. And then there's a dextral form. Dextral is the Latin word for right. So that would be the D or the right-handed form. And so these are molecules that maybe mirror images of one another. So they're not completely symmetrical. They're like opposite mirror images of one another. And so on the left, what we're looking at here, all of that kind of peach colored stuff, that's an extreme close-up of the surface of a protein. And at this point, I honestly forget which protein that was that I rendered. But I actually had a model that showed this small molecule embedded within its binding pocket on the surface of the protein. That's actually ATP right there. This is adenine or adenosine, and then there are 3 phosphate groups down here. So that's adenosine triphosphate or ATP sitting in a binding pocket on the surface of a protein. And on the right, the green outline shows the binding pocket, but here we have a different enantiomer, a different form. And again, I couldn't tell you off the top of my head if one is the left-handed and one's the right-handed, but it's a different form of that same ATP. And notice that only part of the molecule fits nicely in its binding pocket, and there's another part of the molecule that doesn't fit as nicely. So this explains why sometimes the left-handed or the right-handed form of a particular drug works fine, and then the opposite form doesn't work at all. A great example of that is levodopa. Levodopa is a form of dopamine, and it's specifically L-dopa. It's the left-handed form of dopamine. That is the biologically active form of dopamine. The D-dopa, the dextral form or the right-handed form of dopamine, doesn't do anything. And so dopamine is administered to people with Parkinson's disease because Parkinson's results primarily from a lack of dopamine produced by part of the brain. And so you give people dopamine to help with some of the symptoms of Parkinson's, but it has to be levodopa or L-dopa, the left-handed form. All right, so summarizing everything we've talked about so far today about molecular models, we have all these different kind of models that are good for different things. So we have things like the chemical formula, just, like H2O or CH4. And these are great because they easily tell you the relative proportions of the elements. So we know instantly that water is made of 2 hydrogen atoms to every 1 oxygen atom. We have things like the structural diagrams or Kekulé diagrams or skeleton diagrams, which show you which atom is bonded to which other atom, but they don't show you anything about the 3-dimensional arrangement of those atoms. We have things like wireframe models, stick models, and ball and stick models that show you the bonds between atoms and show you very clearly the 3D spatial relationships between the atoms and between their bonds. We have space filling models, also known as sphere models, or Corey, Pauling, and Colton models, CPK models. This uses the van der Waals radius to represent the individual atoms, and it gives us a nice sense for the overall three-dimensional form of the molecule, but the bonds can be hidden. It can be very hard to tell which atom is bonded to which other atom. For viewing proteins, we have specialized models called backbone models or ribbon models, which allow you to easily see the folding of an amino acid chain within the three-dimensional structure of a protein. And ribbon models in particular are focusing on showing elements of secondary structure, like alpha helices and beta sheets. And then finally, we have the solvent accessible model, solvent accessible surface model, which gives a really good sense for the overall three-dimensional form of the molecule. And in particular, it can help reveal the location of binding sites of a protein, these binding pockets where the protein binds to its substrate. So the next thing I want to talk to you about today are different color schemes that are used in molecular illustration, different ways of coloring atoms or coloring whole molecules. So if you go way back into ancient times and back into the Middle Ages, people were already aware of the fact that there were different kinds of chemical things. They didn't really know what an atom was yet. Nobody had really proposed the theory of an atom, but people still understood that there was something called carbon that had certain chemical properties. And people associated certain colors with these things, even back in the Middle Ages. And it was based on some, either the appearance of that substance or some chemical property of that substance. So going way back into ancient times, people have always associated the color black with carbon for kind of obvious reasons. If you understand that things like coal and charcoal and ash are made out of carbon, it's easy to understand why people would associate the color black with that. In the 1700s, much later, some of the gases in our atmosphere were discovered. We still didn't really have an understanding of atoms yet, because that didn't come around until the early 1800s. But in the 1700s, early chemists still recognized that there was something in our atmosphere called oxygen, and they recognized that this was necessary for combustion, for things to burn. And so oxygen became associated with the color red because it was necessary for fire. Nitrogen gas was also discovered in the 1700s. Now, nitrogen is actually the most abundant gas in our atmosphere, makes up about 80% of the Earth's atmosphere. I didn't mention it here on the slide, but for a very long time, nitrogen has been associated with the color blue. Now, I tried to do some reading on this, and apparently nobody really knows exactly why nitrogen is blue, but the best guess is that it's because nitrogen makes up most of the Earth's atmosphere, and when you look up at the sky is blue, except in Rochester. So that may be why people associate the color blue with nitrogen. But it dates back a very, very long time. We just don't know exactly where that originated. In the early 1800s, this is when John Dalton proposed the theory of the atom. He published his first article on the atom in 1805. Now, back then, there was no color printing. And so when you published a scholarly article, like Dalton's article in 1805, he needed some way of representing these things that he called atoms, but he didn't have any color available. So he actually used various kinds of black and white symbols to represent the different atoms, the different kinds of elements. Now, carbon was filled in as a solid circle, again, kind of hearkening back to this association with black coal and charcoal. Oxygen was just a simple circle, hydrogen, a circle with a little dot in it, and you can read the rest of them here. He even had things like gold and platinum and silver and so forth. So he used these symbols in his publications, but no color. Jump ahead about 60 years. In 1865, there was a chemist named August Hofmann who gave a presentation in London known as a Friday evening discourse. It was like the 1800s version of a TED Talk. You get some scientist to talk to a bunch of rich people in a big fancy room. And Hofmann was asked to, give some kind of chemistry lecture, but kind of, dumb it down for the layperson and present this to, a bunch of dignitaries and royalty and rich people. So he gave a presentation called on the combining form of atoms. And it was all about how we were beginning to understand how atoms bonded to one another to make more complex molecules, and how the chemical properties of these different molecules depended on exactly which atoms were present and how they were attached to one another. And he realized that he really needed some visual aids for this TED Talk that he was giving. And so he sent his assistant out into London to all of the sporting goods stores and said, buy up all the croquet balls you can find. They brought these croquet balls back and they used a drill press to drill holes in the balls. And they used little pieces of pipe to connect the croquet balls together. And these things were propped up on these little stands. It's like a little Bunsen burner stand from a chemistry lab. And they would use these pipes to put these things together in various combinations. So this on the left, this is marsh gas, which is actually methane, CH4. This is olefin gas, which I have no idea what that is. I mean, it looks like ethane. It's a 2-carbon chain, but I don't know why it's missing a couple of hydrogens. This is something called Dutch liquid, which I don't know what that is either. I mean, I can tell that it's dichloroethane. It's ethane with 2 chlorines attached to it. Croquet balls come in a limited range of colors. There's black ones, white ones, red ones, blue ones, green ones, yellow ones. I don't know, maybe orange, maybe purple. I'm not sure what other colors there are for croquet balls. But he used some of these colors that had been associated for centuries, to represent the different atoms. So the carbon atoms were the black croquet balls. Hydrogen atoms were white croquet balls. These 2 chlorines here, those would have been green. And there's a simple reason for that. Pure chlorine gas is green. Sulfur, they use the yellow croquet balls because sulfur in its native form is a yellow solid. Oxygen was red because of fire. Nitrogen was blue we don't really know exactly why nitrogen was blue, but he followed these same conventions that had been around in many cases for centuries for these colored balls. And this really then kind of created a standard that has been used in chemistry ever since. When Robert Corey and Linus Pauling created their sets of molecular models, starting in the 1940s, I believe, they followed that same convention. Black for carbon, white for hydrogen, red for oxygen, blue for nitrogen, yellow for things like phosphorus and sulfur, green for chlorine, and so on. And there's a bunch of others. Kind of a reddish brown for iron. It's rust. So here is his model. This is actually the 3D model of an alpha helix on the left. This is, I've shown you this before. That's, my computer rendering of DNA. And the computer software today follows more or less the same scheme. Although I'll show you when we start looking at some of these molecular visualization tools, some of them have some really weird colors, which they just decided, hey, let's just ditch 1,000 years of convention and come up with our own crazy colors, which I don't quite get that. And when you're making a 3D model out of a piece of plastic or a piece of wood, you use black usually for carbon. But if you're trying to represent that on a computer, the downside of using black for carbon is that it's hard to put a highlight and a foreshadow on it. It's hard to make it look 3-dimensional. So most of these computer tools will actually use gray. carbon instead of solid black. So this is actually exported from one of the old molecular visualization apps that I used to use. This one doesn't exist anymore, but it was kind of nice that you could actually export this little table. And that particular software used light gray for carbon, red for oxygen, white for hydrogen, sky blue for nitrogen, yellow for sulfur, orange for phosphorus, green for chlorine, and so on and so forth. And it gives you little color samples here. It gives you the RGB values if you wanted those for some reason. It even gives you the hexadecimal values in case you wanted those. Just a different way of representing RGB. So most modern molecular visualization tools will use kind of a similar color scheme, except for, some of these weird choices that some of them make. Like, for example, there's one application, I think it's Pymol, for some crazy reason decided, let's use bright green for carbon. It's like, what? That's always been the color for chlorine, for hundreds of years. But nice thing is you can change it if you want. Now, there's another color scheme that is used only in coloring proteins. And this is to actually assign different colors to the different amino acids present in the protein chain. Now, this usually only works if we're actually visualizing that protein chain, that amino acid chain. But there's a color scheme here to depict the different amino acids in a protein. And you see the rationale for the choice of colors over here on the right. Turns out the side chain of the amino acid may have a charge to it. That charge may be a positive, negative charge, or alternatively, the side chain may be non-polar. It may not have a charge to it. And so the color scheme is based primarily on whether the side chain of the amino acid is non-polar or polar. And if it is polar, does it have a negative charge or a positive charge? So the non-polar amino acids are represented by dull colors like tan, and this is an unfortunate choice of names to call that flesh because it's obviously only flesh of certain part of our population, but we'll just call it peach maybe. Dark gray, light gray, those are all pretty dull colors. So those would be colors that would be used to represent amino acids that had a non-polar side chain. All the brighter colors are for amino acids that have a polar side chain where there's actually a charge in the side chain. If an amino acid has a negative charge in its side chain, that negative charge always comes from the presence of oxygen atoms. Since oxygen itself is red in the element color scheme, all of the negatively charged amino acids are represented by warm colors. Now, there are two amino acids, aspartic acid represented by ASP and glutamic acid represented by GLU. Those actually have like a full-on negative charge, like a full -1 charge in their side chain. So those get the really bright, vivid red because that negative charge is coming from oxygen. Things like serine and threonine are represented by orange because the side chain is slightly negative, but it's not a full -1 charge. So we use this warm orange color instead of the true bright red for those. There are also some side chains that have either a full +1 positive charge or they may be slightly positive. And we use cool colors for those because the positive charge or the positive polarity is always coming from nitrogen. And nitrogen is blue in the element color scheme. So there are two amino acids, lysine and arginine, that actually have a full charge of +1 in the side chain. So they get this really vivid blue. But then things like phenylalanine, tyrosine, asparagine, glutamine, these get other cool colors like this mid blue and this cyan. Leucine, valine, isoleucine, they get a green. Histidine gets this pale blue. These are all things that have a slight positive polarity, although not a full-blown positive charge. And then finally, there are two amino acids that have sulfur in their side chain. And these two amino acids really have a dramatic impact on the tertiary and quaternary structures of proteins. They're very important for determining those higher level structures. And the two amino acids that have sulfur in their side chains are cysteine and methionine, and they're represented by the color yellow because sulfur is yellow. So you could take a protein, visualize it as a ribbon model so that we're seeing the amino acid backbone, and apply that amino acid color scheme to it. And we're literally seeing each little patch of color represents a different amino acid, and the color indicates whether it's got a positive charge, a negative charge, or if it's a really dull color, that it has no charge, that it's nonpolar. And you can see there's some yellow ones down here, which would be the sulfur-containing amino acids, cysteine or methionine. So there's some bright red ones, which would be things like aspartic acid and glutamic acid, and some of the bright blue ones, which would be the positive charge ones. And then there's some kind of dull gray ones, which are the nonpolar ones. There's another color scheme that's used exclusively for proteins. And I already showed you an example of this, but that's where you represent the model as a ribbon model, and you use different colors to distinguish the different elements of the secondary structure of the protein. So in this case, I used orange for all of the alpha helices. I used purple for the beta sheets, and you can see they even have the little arrowheads on them showing the direction of the chain. And then gray for all these random little loops and coils, which don't really have secondary structures to them. And finally, the last color scheme that I'll share with you, this one is not as universal. The element color scheme, gray carbon or black carbon, red oxygen, blue nitrogen, that's pretty much universal. You pick up any chemistry book, they're going to use those same colors. The amino acid color scheme is pretty widely used, maybe not 100% universal. This one is less well established. I have seen a handful of people who use it. I actually used it for some of my own illustrations, just to kind of try to be consistent with the standard. But it's a set of colors used to distinguish the nucleotides in any kind of nucleic acid. So DNA, messenger RNA, transfer RNA, et cetera, any of those nucleic acids. So there are two purines. Two of the nucleotides are known as purines, adenine and guanine. And what somebody decided was, hey, let's create a color scheme where we take the first letter of the nucleotide or the base, and we try to find a color that matches that letter. So name a color that starts with A. Well, there aren't a lot of normal ones, red, green, blue, purple, orange. None of them start with A. So they said, oh, how about azure, which is just, a kind of blue. So, okay, whatever. Guanine was easy. Green. G for green. Now, I think they picked azure specifically because they wanted a cool color, because I don't know when you would ever do this, but if you wanted to just indicate that one of the bases was a purine, but you didn't want to indicate whether it was adenine or guanine, we just knew it's some generic purine, then you could use like a turquoise color, a blue-green that's kind of halfway between azure and green. The pyrimidines are the other bases, and you see the basic structure of them down here. The two purines are based on this double ring. The pyrimidines are based on this single ring. We'll come back to this much later in the course. I'll give you a whole lecture about nucleic acids too, and you'll learn to love these purines and pyrimidines. Pyrimidines are a little more complicated because you've got the two that you find in DNA, cytosine and thymine, but you've also got uracil, which substitutes for the thymine in RNA. Plus, they also wanted to come up with a color for a generic pyrimidine if you ever needed to show that. So, all right, let's find a color that starts with C. And again, not your basic blue, green, yellow, purple, orange, red. So they came up with crimson. Must have been somebody from Harvard that came up with that, because that's Harvard's color. The T one, somebody really went off the rails for this one. They're like, let's come up with a color that starts with a T. Again, red, green, blue, violet, whatever. None of those worked. And they also wanted it to be a warm color because they still wanted to come up with some sort of generic color for pyrimidine. And since they used crimson for cytosine, they said, hey, let's call it Tweety Bird, which, if anybody remembers, was this little cartoon bird from old, I don't know, Looney Tunes comics. It was a bright yellow bird. So they said, let's call it Tweety Bird Yellow. And then uracil, the only color they could think of that started with a U was umber, which of course is a type of brown. And all of these are kind of in the warm family. Certainly crimson is red, Tweety Bird is yellow, umber is kind of a warmish brown. So they said, all right, let's use orange for the generic pyrimidines. So the most common of these would be the azure adenine, green guanine, red cytosine, and yellow thymine. So one of the molecular visualization applications that we're going to use in here allows you to apply that color scheme to a strand of DNA. So here we see the DNA double helix. It's represented as a kind of a ladder style. We're not seeing the individual atoms in this case, but the ladder is colored using that scheme. So we got our green guanine, we've got our azure or blue adenine, we've got our crimson or red cytosine, and then we got our bright yellow Tweety Bird thymines. And then if we had like an RNA strand in here, like a strand of messenger RNA, all of those yellow thymines would be replaced with brown uracil, umber uracil. So here's one of my illustrations that actually utilized this color scheme. So this is for a physiology book that I illustrated, I don't know, 5 or 6 years ago. And they wanted an illustration to show, the basic structure of the nucleotides. So each nucleotide consists of a base, which the bases are adenine, thymine, guanine, and cytosine. You notice that I actually illustrated the structure of the purines and pyrimidines. So adenine and guanine have the double ring structure. That's the purine structure. And thymine and cytosine have the single ring. That's the pyrimidines. And I used that color coding of crimson, green, Tweety Bird, and azure. And then each base is then attached to a sugar molecule and a phosphate. That's what makes it a full nucleotide. The author of the book didn't request this, but I thought it would be a nice thing to include. I wanted students to be able to understand that there are different ways of representing the DNA molecule. This is just one way of depicting the DNA molecule as a stylized ladder, but it doesn't show you the individual atoms that actually make it up. So what I did is I created 2 different versions of the DNA double helix. One where you can actually see the individual atoms, and the brown part is what's called the sugar phosphate backbone. So that's the stuff that's brown up at the top. That's the sugars and the phosphate groups that are linked together to form the backbone of the nucleic acid of the double helix. And then the bases, the adenine, thymine, guanine, and cytosine, they face inward towards the center of the molecule. And adenine in blue always binds with thymine yellow, and guanine in green always binds with cytosine in red. So there you're actually seeing the individual atoms. But then what I wanted to do is have that fade and transition gradually into the sort of ribbon or ladder type of model, but still using the same color coding. And then over here, we actually show it splitting. And later in the semester, when we talk about nucleic acids, we'll talk about these 5' and 3' ends. Again, hopefully that rings a bell from A&P, that every nucleic acid has a 5' and a 3' end. And the new nucleotides are always added at the 3' end of the nucleic acid as it grows. But anyway, so I decided to use that color scheme to create this this illustration. So that's what I got. So we got different kinds of models, different styles for representing molecules, and each has its own pros and cons. And then we have all these different color schemes for representing molecules.